Thank you. Okay, so our next speaker is Dr. Heyu Ni. Heyu is well known to many of us. He completed his, this crowd. It's the mafia. They've come to take me away. All right. Heyu completed his medical training in China. You'll have to tell me how to pronounce that. Anhui Medical University in China. He ventured into the world of research, did a master's in microbiology and immunology in the same region, and then went to the University of Manitoba where he did his PhD in immunology, and then he was recruited to Boston where he did a postdoc fellowship with Denise Wagner, and it was a fantastic opportunity for him where he contributed major technologies, not to mention knowledge. And then he was recruited to the Canadian Blood Services and the University of Toronto at St. Michael's Hospital as a research scientist there, where he has made major contributions in immunohematology, platelet function, and all sorts of things, sort of out-of-the-box type of science. He's an extraordinary scientist, the most inquisitive guy I know in the field. There's not a meeting where his presence isn't noted by stimulating questions. So it's my pleasure to introduce Heyu to give the next talk.
Thank you very much, Ed, and also I would like to thank organizer, the committee, and particularly Dr. Kim, to give me this opportunity to present here. And I love Vancouver, please. When I first landed in Canada, I think maybe I should spend all my life in Vancouver, but just mistakenly stuck around in the different places. So I do not know whether in the future I could move to Vancouver, but if opportunity open, please do let me know. So as Ed mentioned, I finished my medical training from China, and then did my PhD, and then look around. So my research funded by CIHR, including CIHR Foundation grant, and also Heart and Stroke Foundation of Canada. And I deeply appreciate Canadian Blood Services to give me this opportunity to work in. And also in 2016, I also established a therapeutic company, function slightly different, opposite as compared with Margaret. We try to develop antibody target platelet, try to stop platelet vulnerable factor GPMB interaction, which I believe could be very useful to control the thrombotic event at a high shield condition, such as stroke or heart attack. Maybe also useful for deep vein thrombosis. We will see what will happen. It will move to clinical trials. And I'm working at St. Michael's Hospital, and this is logo of our company. So everybody knows what the platelet, I believe, in the audience. It's very small cell, but the resource that's a little bit argued in the last few years, particularly 2017, once Dr. Mark Looney reported that more than half of the platelet could generate from the lung. So that was, very stimulating, I would say, but subsequently, the globally less well to be accepted, particularly challenged by group from Germany, Dr. Bernard Nussbaum's group, as well as several other groups. So today, we're thinking probably around 10% of the platelet could be derived from the lung. And also, maybe for some of you, maybe do not know, maybe you know, megakaryocytes not only exist in the bone marrow, spleen, but it also able to be identified from the heart, even our brain. So you may think that is really awkward or strange. But the function of these megakaryocytes, whether they generate the platelet, and if they generate platelet, what's the function of them? Whether they're different from the bone marrow resource of the bacteria platelet that has not fully elucidated yet. But in the last couple of years, scientists found that megakaryocytes could have a different way of differentiation, may have a different function of megakaryocyte. Some of them may be major responsibility for inflammation and immune response. Some other working for the hemostasis. That still exists, argument in the world at this moment. So in the last couple of decades, I would, the audience knows that Dr. Jose Lupus and many research in the field, so that platelets are versatile. Not only stop bleeding, but maybe our life through the thrombotic events, but is active contributor for the immune system and acceleration for the atherosclerosis, separation lymphatic circulation system and blood circulation system, and regulate angiogenesis because it contains both pro-angiogenic factor and anti-angiogenic factor. And it has been reported that platelets are friends of cancers and help tumor metastasis. But they also recently report platelet could have opposite action as well. So platelet contribute for the liver regeneration. Some of the surgeons in the audience, you probably know that if you remove 30% or half of the liver, within the next couple of years, the liver resume to the original size. So that is, we call the liver regeneration. That process needs platelet contribution as well. So therefore, platelet has lots of known and unknown functions. So we encourage that the motivated students could involve for this research. Our platelet, the immune cells, this was argued in the last 5 to 10 years, but today I would think people getting tired. If you believe it's immune cell, you believe. So we think that maybe not wrong. Same, if you think it's not, we also need an argue for that. But basically, the platelets similar to their in other thrombocytes in the low level of vertebrate bred animals, that do have immune functions, such as they contribute for inflammation, help leukocyte migration in vivo. Many of you knows, or many of you observed, platelet leukocyte heterotypical aggregation. That is contribute for inflammation and the leukocyte migration. Platelet also involved for inflammation, infections, platelet-bacterial interaction. If some of you did you heard some research from Utah previously? If you co-culture bacterial and platelet, you could see the both sides try to develop a weapon to hit each other and protect themselves. Platelet could also involve for the infection of virus, even parasite. And some of you, and also our Canadian Blood Services, aware about that malaria-infected red blood cell that could easily to cause the platelet to stick with it. So that's leading to apoptosis of malaria. So this paper published in 2009. In 2012, the same group published the paper in Science, demonstrated that apoptotic factor released by platelet is platelet factor 4. And the platelet also contribute for the lymphocytes homing, so that work was done in the early time by Uli's group in Harvard Medical School. And probably even more strikingly, actually, this almost terminated my CIHR grant because I have exactly the hypothesis regarding this, because platelet contain CD40 ligand and CD40, which is essential for immunoglobulin class switch. Everybody in the audience do have IgM, and also IgG and different other type of immunoglobulin isotypes. So our immunoglobulin switch original from IgM, switch to IgG or IgA or IgE. So that is, we call the immunoglobulin class switch. But FAB, the variable region is the same, but then the loop of other, the mu chain out, switch to the gamma chain, that will become IgG. So this process do need CD40 ligand. So that to be beautifully demonstrated by the group, original in Iowa, but now they moved to the Purdue University I forgot the state, excuse me, but moved to the Purdue University. So that clearly demonstrated that platelet, if you transfuse to CD40, the knockout animals that could drive the immunoglobulin class switch, and also to facilitate germinal center formation that is placed to generate antibody. And we still puzzled a bit, so why platelet contain large amount of TGF-beta, which is potent immune suppressive cytokines. So the information just gradually appeared out. And also Dr. John Sample original from the St. Michael's Hospital, he also wrote a review article regarding platelet and the interaction with immune systems. Today we're thinking platelets are part of innate immunity, and through the different mechanism, or even we not fully understood, contribute for adaptive immunity, interaction with T cell, B cell, dendritic cell, maybe other cells, so affect adaptive immunity. In the 2009, we published the paper in Blood because of what we found that in the fibrinogen deficient animals, if you originally we tried to study whether fibrinogen von Willebrand factor deficient animal, the platelet more easy to activate it. But my postdoctoral fellow told me it's opposite. It's not. I said that's going to be strange. If without fibrinogen von Willebrand factor, platelet should has certain level of compensation to better has a hemostatic activity, stop bleeding. So I would assume in platelet should be more easy to be activated. But why my fellow told me is different, is opposite, because what she found is the P-selectin expression in this platelet is less. So because P-selectin expression CD62P is a golden marker for platelet activation, but that's expression less. So she's assuming that is less active for this platelet. But subsequently we found it's not. Because of platelet less activation, because of platelet has less P-selectin, because fibrinogen and beta-3 integrin interaction that could drive residue mRNA of P-selectin de novo, newly synthesized new P-selectin. So therefore P-selectin level is higher in the regular platelet, but not in fibrinogen deficient platelet. And we also further demonstrated that fibronectin has similar effect. So therefore, that after fibrinogen transfusion, maybe not only just affect our hemostatic factor, but probably also the immune responses, because P-selectin contribute for the Th1 immune response. So that's, today we do not know, but I would encourage you, if it's possible, to check after in a human patients after fibrinogen transfusion whether the immune response changes. So this is my students wrote a review article regarding this two Asian system, immune system and the hemostasis system that has interactions. So generally, we believe or we think in platelet are pro-inflammation and pro-immune response, as what I listed there. But it's very surprising, once we started immune response against the platelet GPMB alpha, because we found anti-GPMB alpha usually less immune responsive as compared with to GP2B3A. We suspect that probably because of platelet GPMB alpha has lots of sugar on it. That's Dr. Karen Huffmaster very well about it. We're thinking probably because of the sugar decoration on the GPMB alpha surface that's prevent immune system MHC presentation. As we all know, MHC does not present sugar, the peptides, and does not express the lipase-related peptides, the only peptides. So therefore, we're thinking if we get rid of this sugar, probably could enhance immune response. That's the original idea. So then we decylated this platelet, so then transfused to the animal. But what we found is another opposite observation. We found that immune response against decylated GPMB alpha, the antibody generation is get less. And not only GPMB alpha, the beta-3 integrin, the same thing. You get less immune response as long as you go with desylated platelet. And even more, so if you say that is a platelet antigen, so sheep red blood cell is completely different from the platelet antigen. If you co-transfused with desylated platelet, the immune response to sheep red blood cell antibody level also decrease. And even more interestingly, if you give that factor 8 to express in the platelet, that platelet desylation, this immune response against the hemophilia A-like factor 8 immune response also decrease. So therefore, this may be not only just immune response, but also has a significant translational value. So that means I would think in probably every day, daily best, old, aged, activated platelet, desylated platelet, clearance in the liver control our autoimmune response, protect us from autoimmune disease. That is what we try to mention here. If you're interested, you probably could read this paper published in Science Partner Journal. What happens here? We found that the desylated platelet, if you transfuse to the animal, they not come to the spleen, but come to the gut and also come to the liver. Okay, so they exclusively come to the liver, but not the spleen. So this is confirmed by flow cytometry. We pick the different organ, the liver, lung, and the spleen. We found normal platelet, the desylated platelet will stuck in the liver, but normal platelet could be in the lung. Some of them stick there, so majority of them may go to the spleen. So then we found that the Kupffer cell, the macrophage in the liver, so that's control for this platelet clearance. If we use a coordinate, you could see stop this platelet clearance. And we found that both this GPMB alpha required, because if you get rid of GPMB alpha, this will not occur. This will not occur clearance in the liver. So then, we further started this cytokine file. We found that desylated platelet could increase the anti-inflammatory cytokine IL-10 and also TGF-beta, which is dose-dependent. If you give more desylated platelet, so this cytokine, anti-inflammatory cytokine level will increase. And even more, we found this could affect not only just the local, but systematically, because the T regular cell that could migrate to the everywhere. So we found that desylated platelet transfusion, you could also increase peripheral CD4 positive T regular cell, and also in the spleen, this CD4 positive T regular cell level also increase. Again, if you shut down the Kupffer cell, this will not occur. So therefore, we propose that once the platelet activated or cleared, so that through this mechanism, the liver clearance induces immune suppression, this process, and this is systematically. And another report from Craig Morrow's group, they also found that resting platelet could interaction with monocytes that could also induce immune suppression process. So therefore, at least the two mechanisms that demonstrated platelet could anti-inflammation and decrease immune response. So therefore, we think probably between this resting and aged condition, they may contribute for the promotion of inflammation and immune response. That we mentioned platelet regulate the immune system and also attenuation of immune response. Immune system could also target platelet, as most of the scientists, clinicians, and students understand that immune thrombocytopenia is the immune system target platelet. So one of the diseases is mother during the pregnancy that has immune response against the fetal platelet antigen. So this antibody from the mother, so then cross placenta target baby that could lead into thrombocytopenia. So this disease is a little bit more severe as compared with ITP because significant intracranial hemorrhage occur in the babies. So this mechanism, as what I mentioned, that's a fetal platelet antigen triggering the mother immune system through the neonatal FC receptor. That is also our group to identify that. The neonatal FC receptor in the placenta, particularly from the baby side, not particularly, from the baby side, is responsible to transport mother IgG across the placenta to the baby side. If you stop this process, you may be able to ameliorate this disease. So this work was published in 2010 for this. So in the Caucasian population, most of the antigen occur in the PSI domain. So leucine, so the polymorphism that's replaced by proline, that could lead to this anti-human platelet antigen 1, this immune response. That is the major in the cases, reported cases in Caucasians, more than 80, 85, or even 90% of this disease caused by antibody against these polymorphisms. Mother, so father, the amino acid difference that's triggering the maternal immune response against that. In the Asians, maybe targeted to this I-like domain, or against the CD36 is sort of dominant. And it's strange and weird that has a little bit antibody against GPMB alpha. So that we call the human platelet antigen 2. The reason we not fully understood, but the reported few cases showed the symptoms are pretty severe. So in the last, I would think, couple of decades, we established the first animal model of fetal and neonatal alloimmune thrombocytopenia. We demonstrated that gamma globulin transfusion is useful for this. At the time, once we published this paper, that's argument. In the US, people say that's New York phenomenon, seems to IVIG works. But in European, they feel it does not working. But after we published this blood paper, we showed clearly IVIG is useful. Then seems to me the European, the concept slightly changes. IVIG is useful to control this disease. I would think is decreased. The mechanistic is still complicated, but one mechanism could stop the neonatal FC receptor transport the maternal pathogenic antibody cross placenta. We found FcRn is important for this, and we found that anti-GPMB alpha antibody, if you got it, I would think probably Dr. Joselupus will sort of agree with me, antibody target GPMB alpha cause platelet activation. But this activated platelet may generate pro-coagulation situation in the placenta, so therefore thrombus formation in placenta, mother antibody, mother nutrition cannot cross placenta to the baby. So we found that more than 83% of the mice, the female mice, pregnant mice, not able to deliver any babies because each mice could deliver 6 to 12 pups, but we found more than 83% of them, no any baby could be generated. So most recently, that's you said that's animal model. Animal has a different placenta as compared with human. Recently in the in Toronto, there is a 3 frequently miscarriage woman. We found 2 of them has antibody against GPMB alpha. So I do not know. I encourage clinicians further explore whether this is true in a human. We also found that anti-angiogenic effect, because you bonding to beta-3 integrin, could affect angiogenesis. So that's probably the major reason to lead into the brain intracranial hemorrhage. Because in the baby, in the fetus, everybody, particularly clinicians, you know, once we deliver baby, if the brain come out, so the whole body easy to come out, right? Because the brain in the fetus condition has very massive angiogenesis in the brain. So maybe anti-beta-3 integrin, particularly against alpha-V beta-3 integrin, impaired the angiogenesis. That could be the reason leading to the intracranial hemorrhage in this disease. So this is also has diagnostic value for the screen, the mother, whether the baby has more prevalence for the intracranial hemorrhage. We also found the NK cell urine at the early stage in placenta is good for the angiogenesis of placenta. But if NK cells stay there or not move out, that could generate the inflammation leading to miscarriage. Another immune system to target is to target ourselves. We call the ITP, as we mentioned here. So about 70 to 80% of ITP autoimmune antibody target fibrinogen receptor, alpha-2b beta-3 integrin, or we call the GP2B3A. So this signal pathway is complicated. This is from Dr. Sandy Shetil and Dr. Peter Luhmann to draw this picture. Another potential autoantigen is also von Willebrand factor receptor, as we mentioned. If the mother has antibody against this, that could be very dangerous. Maybe you're not able to generate a baby or deliver living baby. In the autoimmune thrombocytopenic condition, about 20 to 40% of ITP patients have this antibody. And some of the patients has antibody against both. Has antibody against GPMB alpha, also has antibody against GP2B3A. Some of them. But one of the study, through collaboration with my Chinese colleagues, we found that no matter you have anti-GP2B3A or not, as long as you get an antibody against GPMB alpha, you will not sensitive to first-line therapy, steroid, as well as not sensitive to IVIG therapy. So that has a significant difference. So what's the mechanism of that? Dr. Bonaniswan's group found that in the early years, about 20 years, 25 years ago, report that if you have antibody against GPMB alpha, if you get rid of FC portion out, you still cause thrombocytopenia. But if the antibody against GP2B3A, you cut FC portion out, they cannot cause thrombocytopenia. So therefore suggests that anti-GPMB alpha may have significant difference from anti-GP2B3A. Anti-GPMB alpha may need an FC portion, means FC independent thrombocytopenia. Therefore, because I'm working for Canadian Blood Services, we want to rescue or decrease the IVIG abusement, let's say that way, because it has a national-wide shortage supply or shortage resource for gamma globulin, IVIG. So therefore, we hypothesize that if GPMB alpha is FC independent, maybe IVIG does not working well with them. So subsequently, we did publish a paper in Blood, found that indeed, we test 5 antibody against GPMB alpha. Only one of them responds to IVIG therapy, but test 5 or even more anti-GP2B3A, they all respond to IVIG therapy, okay? So that's all this data is consistent. And also in China, in the nationwide ITP patients, they found IVIG indeed less responsive in these patients, and also less responsive to steroid therapy. What happens? So then we found that this mechanism to cause this FC-independent platelet clearance is because of platelet desylation. That time, we heavily influenced by Dr. Karen Huffmaster regarding this platelet desylation. We found that once antibody binding to GPMB alpha causes platelet activation, caused neuraminidase translocation from alpha granules to the surface, leading to GPMB alpha desylation. So this desylated platelet be cleared in the liver, but not spleen. And the platelet cleared in the liver maybe is one of the previously less evaluated, less appreciated, less recognized mechanism. Because platelet, every day we have lots of aged platelet, they may be all cleared in the liver, but not in spleen. I open this for the challenges, for the evaluations. So we further found that if you platelet desylation is correlated with first-line therapy. Get more desylation, maybe less, not maybe, in this human ITP patient is less responsive to the first-line therapy. We're thinking that if it has antibody against GP2B3A, you're relatively sensitive to steroid or IVIG therapy. this platelet cleared in the spleen. However, if you get antibody against GPMB alpha, no matter with or without anti-GP2B3A, so this platelet distillation will occur, clearance in the liver will occur. I would like to mention that some of the antibody against GP2B3A also leading to platelet distillation, because human platelet different from mouse that has Fc receptor. Once antibody against GP2B3A, Fc portion engaged with Fc receptor on the platelet surface, this cross-linking could cause a platelet activation and a neuraminidase translocation. This platelet may also clear in the liver. So this group of the patients is less sensitive to first-line therapy. And recently there is some new information suggests that the thrombopoietin and also mimetics, including anti-CD38, this is very new therapy, looks like they're all a little bit difficult to handle this type of patients. Hopefully, these events not only just from China, not only just from Europe, hopefully that could be in North America. Not necessarily hopefully, but I want the patient easy to be treated. That's basically. So this work, the immune system was mainly studied by my PhD student, Jun Li. And so my the fellow will present regarding the most recently our paper published in PNS regarding the thrombopoietin production from this clearance process. So Grace Wu is in the audience, and also Slegina in the audience. Slegina will present something related with blood coagulation. So this work cannot be done without collaboration with our local at the University of Toronto and our collaborators internationally, including those from China. So this is our group. So that's immunology study is from these students, and the TPO generation is from this student. These two ladies, some of are my trainees working for the company regarding the drug development against GPMB alpha. So this monoclonal antibody, we generate a large amount of monoclonal antibody. We generate around 100 strands of monoclonal antibody against the different platelet proteins. So Slegina in the audience, she's a hiding hero. So hopefully she will able to present her information for you. So this is Toronto. It's not too bad, but compared with Vancouver, I would still prefer Vancouver. Thank you. Thank you.
I didn't know that was Toronto. That's not Toronto. We have some runners. Yeah, right beside you.
Thank you.
Jose.
Thank you for, is it on? Yeah. Thank you for that talk. I have a couple of questions. Maybe I'll just ask one. So in the Kupffer cell-mediated clearance that involves GP1B alpha, can you describe what you think the molecular mechanism is? Is it direct, what the receptor on the Kupffer cells is, and whether or not it needs an intermediary protein like von Willebrand factor?
Mm-hmm. Well, this is a really good question, and we try to address it. So, but we did find that antibody block von Willebrand factor A1 domain interactions may be able to attenuation at least, if not inhibit this process. But we do not know whether GPMB alpha von Willebrand factor direct interaction that's mediated Kupffer cell clearance. That we have not fully elucidated yet. But clearly, this process is involved.
I guess some of those antibodies might also, if they're against GP1B alpha, might also block other interactions, like the interaction with alpha M beta 2 integrin.
Yes. So you mean that such as MAC1, and also we appreciate your work in JXMAT regarding that probably also able to block P-selectin. So that could significantly affect this leukocyte, including Kupffer cell interaction. Mechanistic, we still not fully understand, elucidate yet, but I would think probably multiple interactions, tangle between them may be involved.
Thank you.
Thank you for the great talk. I have a question about like the selected platelet and also the regulatory T cell population. So when you adopted, when you put the selected platelet in, and you see the increase of the Treg, do you know the Treg also showing increase their function like suppression or cytokine production? And also, can you provide some insight how does it change this Treg increase because it affects their differentiation through DC or some cytokine? Thank you.
I hopefully you are the CIHR grant review. That is a question we try to ask for many to figure them out. So we not fully understood, but clearly after Kupffer cell uptake, decylated platelet itself, they generate more, the cytokines, may also affect the local inflammation process. So clearly, so that's because platelet has TGF-beta, so that's probably TGF-beta somehow to drive the T regular cell differentiation development. So locally in liver, probably could also systematically, but it's unlikely very far because TGF-beta we consider is a relatively local cytokine because easy to stick in the local around, right? We try to figure out, so that is one of the major projects, the GRACE, try to figure them out, how these drive the T regular cell come to the peripheral, come to the spleen, right? Is more T regular cell generated or more T regular cell differentiated, or is more T regular cell from hiding area released to these places? We still not fully understood, but we try to figure them out. We need money, so that's a big problem.
Hi, Heyu. A little bit of a transfusion-y question. I'm curious if cold-stored platelets are behaving similarly to decylated platelets in terms of the organs to which they are trafficked, because I do know that cold-stored platelets are not the same.
Mm-hmm. Yeah, thank you very much, actually. So that is what I ask Canadian Blood Services to patent this finding. So I propose that, so if you cheer the platelet, as Karen Huffmaster to show that earlier, so that could induce platelet activation and also the decylation. So whether we could treat ITP patients, use their themselves platelet. Of course, ITP patient already has a limited platelet, but whether we could use other blood donor to give the platelet, put them, cheer them, then transfuse it to these people, could quickly induce the immune suppression. I try to figure them out, and I ask CBS to give us some small amount of money to do the pioneer test for this. Thank you very much. That's a very good question.
Thanks.
Thank you.